All right. Hello, everyone. My name is Alexander Shu, and today I'll be discussing hyperparasitism as the special topic for the Microbiology 580 course at Oregon State University. So this presentation focuses on the topic or the um, the statement, which is the exploration of the diverse strategies of hyperparasitic organisms, excuse me, and the ecological effects they have, and understanding the emergence of these complex and highly niche relationships. So we're going to look at hyperparasitism. We're going to define it first, and then we're going to look at three focused examples of hyperparasitism before moving on to the discussion on ecology and evolution of hyperparasitism before concluding the presentation. So firstly, starting with hyperparasitism. The easiest way to understand hyperparasitism, or what a hyperparasite is to understand that a hyperparasite is a parasite of a parasite. It's a little hard to understand, but there's an image here that's been provided which showcases this superhost, the one that's the largest one, and within that we have a secondary host or secondary parasite, or excuse me, primary parasite, which is a secondary host, and then the secondary parasite, which is the hyperparasite. So it's when parasites themselves have parasites. Um, they the research that was able to be done on hyperparasitism, they looked at a lot of papers in order to make this presentation, and it was implied mostly that hyperparasitism or hyperparasites arise similarly to parasites themselves, which is they find a niche instance in the wilderness where it is much more expedient as a survival method and a method of propagating their genetics and their offspring. It is much more expedient to just use this other organism that happens to be a parasite. That's how hyperparasitism is uh, implied to have been arised. Um, they also serve as biocontrol agents themselves because hyperparasites can control the populations of parasites and indirectly the populations of hosts or host organisms. That's why there is the possibility of phage therapy, which is, it's not necessarily the same thing because phage therapy is when you use bacteriophages to attack bacteria to control bacterial infections. And this has been in this paper from 2019 indicates that there is some utility to the method. This picture here is the genus of Diptera, which is one of the common organisms, mostly they're flies, that are the hosts of these hyperparasites. So they themselves are mostly parasites in many instances, but they can also serve as hosts to hyperparasites. And a topic that we'll tie in later, but it seems appropriate to mention here is the idea of hitchhiking, is where both the parasite and the hyperparasite are transmitted together to a host. It's mostly a vertical method or it's vertical as where the offspring of a hyperparasite is transmitted to the offspring of a parasite, and perhaps both of those are then transmitted to the offspring of the host. It's a little complicated, but if you need to rehear that statement, you are more than invited to. So the first focus species or focus hyperparasite that we're going to look at is the genus of Label benialis. Label benialis, it's a little hard to say. So these are a group of fungi that are found in the caves of South America, say Brazil, Bolivia. I think there were some in Ecuador as well. Many of the South American continent nations, they're found there. In order to understand, I'm going to start with the parasite, which is the bat flies of the genus Diptera, Nycterbiridae, and Strepsilidae parasitize Chiroptera, which is bats. So these bat flies parasitize bats. The fungi of the Labobenials genus Orthorhynchus, Gloeandromyces, and Nycteromyces attack the bat flies. So we have this wonderferful paper here. Where's my mouse? Here's my mouse. We have this a section, it's an excerpt from this paper that was used in this research for the presentation, where we have the bats being parasitized by bat flies, and in turn they're parasitized by hyperparasites. I, in my research, was not able to find an instance other than this one where hyperparasites is spelled with an HI, but it could be it's just possibly a different nation's way of spelling it. But it is a hyperparasite in this row, on the very most right row. What was able to be garnered from researching this topic on labeled benials is that there is prey specificity where certain hyperparasites will target specific bat flies to propagate themselves as well as their offspring, but there are instances where transmission to atypical hosts is possible. It does occur where, for example, an organism or a labeled benial gets transmitted to some other bat fly it usually is not found on. That does occur, but it's mostly rare. So continuing the discussion, here this picture showcases the bat fly. They don't look like the flies that normal humans interact with, but these are indeed bat flies. And we have a picture here in the upper right corner of hyphae growing out of the thorax of the bat fly itself. So this is the example of the hyperparasite themselves that are actively taking nutrients. So label beanials are lethal parasites. So according to this 2020 paper that we have cited uh, right below this statement, it was stated that half of the survival rate of the bat flies in question was efficiently cut in or effectively cut in half. Half of the population was uh, attacked by labeled venial fungi, and they were able to stop that much of the population from continuing to live. So they are very efficient and pretty lethal parasites or hyperparasites. Parasitism of the fungi themselves arose at least 3 times independently, according to this 2018 report. Uh, in the Eastern Hemisphere, Arthrohanchus arose as the single group, and in the Western Hemisphere, Gloea andromyces and Nycteromyces arose. And this ties into the idea how hyperparasitism itself, because it evolved independently 3 times, it seems to be a very efficient and it's a utilitarian method or lifestyle for organisms to propagate themselves. So that'll tie in a little later when we have the discussion on hyperparasitism, ecology, and evolution. Currently, there are 14 species of fungi that have been associated with 19 species of bat flies, and in turn, 15 Chiroptera species. So it's a very large network of hyperparasite, parasite, and host organisms, but further research is probably useful for understanding this concept, or at least this specific instance more. The second um, hyperparasite focus species or instance that we're going to look at is Lysibia nana and Gallus agilis. So the parasite, or excuse me, the caterpillar Pyrrhus brasisiae is parasitized by Cotetia glamata. So it's a caterpillar that's parasitized by one species of wasp. But in turn, Lysibia nana itself as another wasp, it's a hyperparasitoid. So when a word has toid at the very end, it usually indicates some kind of wasp, a wasp creature. So Lysibia nana is a hyperparasitoid. The hyperparasitoid of Lysibia nana attacks the prepupae of Cotetia. So it means it attacks the non-pupae. So pupae is a chrysalis. So it means it attacks the larvae or yeah, the larvae that has not become a pupae yet, that is in the caterpillar host. There is an 80% success rate for the hyperparasite. So they are very efficient hunters when it comes, or hunters of parasite hosts and possibly hosts themselves. These are found mostly in European countries. One of the reports indicated that it was found in France and warmer Mediterranean or European nations that bordered the Mediterranean Ocean. There is strong coevolutionary evidence present in the ability of the wasp to find cocoon broods of C. glomerata within caterpillar hosts. There's many ways that this has been observed, but one of them is the odors from the caterpillars themselves can help indicate which caterpillars themselves have been parasitized by uh, C. glomerata. And another one is that the plants also seem to release a specific marker or some kind of, not necessarily a pheromone, but some kind of chemical is released. So this diagram here was taken from one of the sources. The specific image that should be focused upon is the left or right lower one, because it is a Lysibia nana targets the prepupae larvae of C. Cotesia glomerata. So they're injecting their eggs into the larvae while it's still inside the caterpillar host. So that's the image you want to focus on. It just so happens that there are many other ways that many other wasps are able to find larvae at different or find their parasite hosts at different instances of their life cycle, but the focus one is the lower right one. The hyperparasitoid wasp, Lysibia nana, seems to be able to utilize herbivore-induced plant volatiles, so some kind of chemical that is released by the caterpillars to target the caterpillars that have the Cotesia glomerata larvae. This is a very interesting concept because it showcases that the hyperparasitoid is not necessarily paying attention to where the wasp parasite, the parasite that it's looking for, or parasite host, it's looking for what the host emits in order to find the hyperparasite because it's not looking for just a host. It's looking for the host plus the parasite. So there's strong, this kind of mechanism indicates a strong coevolutionary and very intimate evolution between the three species, actually. And moving on to the third and final focus species or focus species instance is Dothidiomycetes and Sardireomycetes. Very long words. These are also fungi, and the parasite in this instance is black mildew, which is Meliolales, and the Meliolales as black mildew attack vascular plant parasites. There was not, during research on this topic, it didn't seem to indicate that black mildew species targeted any particular plant and creature, just that they targeted they targeted vascular plants in general. So they are very broad species, or they can attack a broad uh, range of hosts as parasites themselves. And the two fungi species of hyperparasites can prey on the black mildew. This image here, so it shows first the large picture, and then it zooms in from A to B to C, where A is the biggest picture, and then B looks at a small colony of the black mildew being colonized in turn by the hyperparasites. And picture C showcases how the yellow fungi are attacking the hyphae of the black fungi on the leaves. So the the hyperparasites are either necrotrophic or biotrophic, which means two different things. So necrotrophic, necrotrophic means that in order to acquire the nutrients found in cells of other organisms, it has to kill those cells or in a sense, rupture the cell, which is kills the cell. In the process, they have to rupture and kill the cell in order to acquire the nutrients. Biotrophic, conversely, means that the cell that contains the nutrients does not have to be destroyed, or the cell itself does not have to be the cellular membrane does not have to be penetrated in order and destroyed, and thus killing the cell in order for it to acquire the nutrients. So it doesn't kill the cell to acquire nutrients. These two hyperparasites are milder, so they actually don't penetrate the black mildew cells, though some of those variety have been discovered. So focusing more, there are 535 hyperparasitic fungi that have been observed worldwide according to this 2022 report. And this image here showcases the different types of shapes or different morphologies. That's a biological phrase, morphologies, which means the shape of an organism, that are taken by these wide ranges of hyperparasites, and it seems to change depending on which species of black mildews they use as hosts. The problem with this specific focus species, though, and hyperparasites in general, is that hyperparasites are extremely niche, and particularly in the case of hyperparasitic fungi that parasitize black mildews, it is not fully studied yet. Even right now, it's 2024. This study came out in 2022. It's not really fully studied, but hopefully more understanding will be available as research progresses. It has been noted that they are by the same 2022 paper, these are two different ones, but from the same year, but it is a potent population controller of black mildew. Now moving on to discussion of ecology and evolution of hyperparasitism. So hyperparasites are highly attuned to not just the parasite, but the host health as well. We saw this in the instance with the Cotesia glomerata and the caterpillar and Lysibia nana as the hyperparasite. Lysibia nana is able to look at the health, which is the caterpillar, the health of the host, and observe that the host is emitting specific odors due to the parasite that has in their body, and is able to observe that. And it also makes a lot of logical sense because when you think about it, if a hyperparasite is going to find a parasite, it's not just using the parasite itself, but it's using the body indirectly of the host, which contains both of the organisms. And if you're not careful with keeping not just the parasite, but the host itself alive, you're not going to really be able to propagate yourself. Hyperparasites also directly influence parasite virulence. So we have two sets of graphs A and B here from one of the studies that was cited in this research, and it showcases how, depending on the virulent and strength of the hyperparasite as well as the strength of the parasite, how they will be able to propagate together in the host. So graphs A indicate that there is slow coevolution between the parasite and hyperparasite, which means that they're not really responding to one another. They're doing things that really have nothing to do with one another. And then looking at it from the perspective of a hyperparasite, looking at this graph here with more color for square A, there initially is a lot of hyperparasites, or excuse me, there's essentially no hyperparasites before it peaks and then it goes away because there's no strong coevolution. They're not evolving together. That's why the hyperparasite dies out. It means the hyperparasite is not responding to what the parasite does. However, in the case of instance B, where hyperparasites and parasites are both responding to one another, um, there eventually there's going to be the hyperparasite and parasite population, which is indicated by H. So in this case, or in this study, H indicates there is a host, a parasite, and a hyperparasite. S indicates just a susceptible host, so the susceptible host doesn't have any parasite or hyperparasite, while P indicates hosts and parasites, so an organism that is serving as a host to a parasite. It clearly shows that when they are able to respond to one another, where the parasite and the hyperparasite can respond to each other's virulence, there is more of the hyperparasite present. And what I just mentioned is the key detail, which is, again, the parasite and the hyperparasite virulence tie into one another because according to this research that was done and this paper, if the virulence of the hyperparasite is stronger and higher, the virulence of the parasite is also going to increase. And the reasoning behind this is as such. A hyperparasite being virulent means that it's able to find more and more parasite hosts and kill more parasite hosts. The parasites themselves, in order to respond to being killed off more, have to make it up or have to make up that loss by being able to find more hosts themselves, which increases their virulence as well. It is noted, however, that many parasites seem to be able to increase or decrease their virulence depending on the population and virulence of their parasite hosts as well as the hosts themselves. So that's where the idea of the hyperparasites being highly attuned to parasite and host health ties in again. And the overall host population increases in the instance where there is hyperparasites. The reason for this is because hyperparasites are biocontrol agents, which means that they are able to control the population's health and virulence of the parasite organisms, which are somewhat pathogenic. So that's why they are biocontrol agents. And if they control biocontrol, or if they are able to mitigate biocontrol agents, that's easier to understand, mitigate parasite populations, they are therefore able to allow host populations to increase because the parasites serve as pathogens, so they prey and sometimes, maybe very often, kill host populations. But if they themselves are mitigated, the hosts are allowed to propagate more, which is why host population increases. Moving on to the second page of discussion. Hyperparasites serve to keep different populations in check. So this includes the hosts themselves, um, but also, and obviously the parasites, because that's the population they're preying on. The host population um, recovery rate is showcased in these graphs here, and I can go through each of them in turn. So looking at graph A, where hyperparasite virulence is um, on the y-axis, while parasite virulence is on the x-axis. This ties in again to how when the hyperparasite is more virulent, the parasite, in order to make up for the fact that they are now being preyed upon, themselves has to be virulent. So that's why hyperparasite virulence drives parasite virulence. So this is the co-evolutionary idea where that parasites and hyperparasites, once they come in contact with one another, they evolve together, and it's to the benefit of not necessarily the parasite, but the hyperparasite that this occurs, because then they can respond to the parasite and be propagated more, showcased in graph B of the slide before this one. And fungal infections control bat fly populations. This is a great example. Again, they were able to mitigate the population of bat flies by 50%. That is a very, very strong, um, not necessarily predator, but as a biocontrol agent, that is a very efficient and strong biocontrol agent. Moving on to graph B here of the hitchhiking probability. So this is where it ties into how when viral or virulence is stronger, um, and the probability of being able to propagate themselves to the next host goes down. This is because if virulence is strong, that means they killed their host. And the host of a hyperparasite is a parasite. They need the parasite to be alive so that the parasite can then propagate offspring to the next host, and alongside that, hitchhiking the hyperparasite themselves. So this is where the trade-off becomes quite very important, very critical, and why hyperparasites would be inclined to perhaps decrease their virulence, because if the virulence of the hyperparasite is too much, their hitchhiking probability goes down. So they have to balance those two. Host recovery rate is a little harder. I think the paper said that, if I remember correctly, the paper said that host recovery rate for this instance was a little harder to detect, but there was some evidence to suggest that when the virulence of a hyperparasite is extremely high, the host recovery rate goes down because the hyperparasite virulence drives the parasite virulence. So indirectly, the hyperparasite being more virulent will end up killing the host which is why they're not able to recover as a population. However, with the host being able to recover, that would occur usually when both the parasite and the hyperparasite, because they move alongside one another when it comes to the virulence. If both of their virulence is down, or let's just say not very large compared to other instances that we see on the left side of graph C, if they're very low, if both the virulences of the parasite and the hyperparasite are low, that indicates that the host would be able to recover more because it's not suffering as much from the parasite and indirectly the hyperparasite. So concluding this presentation, hyperparasites inhabit distinct niches globally. There were 3 of them that were able to be looked at. In case I didn't mention it, the fungi, the black mildew hyperparasites, those are found in a wide range of countries. They're found in India, China, many South American nations like Bolivia, Ecuador, Bolivia, and Brazil. Um, so there's many instances of these globally. They prey upon distinct parasite hosts. They don't prey upon hosts themselves directly. They prey upon the hyperparasites. This picture here is a large image of Lysibia nana, or no, this is not Lysibia nana, but it is one of the hyperparasitoid wasps. In this case, it's a hyperparasitoid wasp that is looking for the pupae, because Lysibia nana looks for prepupae. So this is not Lysibia nana, but it could be perhaps a different species of the same genus. They serve roles as biocontrol agents because they are able to mitigate the populations of of parasites themselves being hyperparasites, and in turn host populations. And they can drive forward evolutionary intimacy because as the virulence of the hyperparasite and parasite become, say, larger, they're going to need each other in a sense, because if the virulence of the parasite becomes too large, then they're going to kill off the host, which is where the hyperparasite virulence comes in to perhaps take away the population of the parasites, so then allow the host population to rise again. So again, evolutionary intimacy is key to hyperparasitism. They're pretty much locked into this relationship, and it's probably going to be very hard for them to find another instance or another ecological niche. However, there is much more research that needs to be done on this topic, largely because there's frankly just not many available instances. Maybe there's not enough research, but it is a very interesting topic that is probably, I believe, worth a lot more research and looking into. Here we have reference page 1, and here we have reference page 2. Okay, well, that was the presentation on hyperparasitism for Microbiology 580. Thank you for your time.